Have you ever tried to open one of those impossible hard plastic clamshell packages without breaking what's inside? Well, imagine trying to do that, but on a microscopic scale. That's essentially what we're looking at when we try to extract the literal code of life, DNA, out of a cell. It is exactly like unboxing a heavily fortified package. We've got to get through those tough outer layers, clear out all the complex packing materials, and do it all without damaging the fragile prize hidden deep inside. It's an incredibly delicate process, and honestly, it's the absolute foundation of modern molecular biology.
Welcome to this explainer. Today, we're diving straight into the fascinating science of nucleic acid extraction. We're going to explore the massive engineering and chemical hurdles involved in taking what used to be a highly sophisticated multi-step laboratory process and shrinking the whole shebang down into a portable device. We're talking about taking science from the lab bench directly into your living room or right to a patient's bedside, all without losing an ounce of precision.
Here's our roadmap for today. First, we'll cover the basics of extraction, check out the traditional chemistry set, and look at the solid phase revolution. After that, we'll shift over to point-of-care devices, tackle a pretty dramatic engineering conflict where, spoiler alert, chemistry actually melts plastic, and finally, we'll wrap up with the future of diagnostics.
All right, let's jump right into part one, the first step, extraction.
So here is the core biological problem. Before we can sequence a genome or identify a virus or run any kind of diagnostic test, we have to actually get our hands on the intact genetic material. And this is where the magic of extraction begins. It brilliantly breaks down into 4 universal steps. Think back to our unboxing metaphor for a second. Step 1 is cell disruption. Basically, just breaking open that cardboard box. Step 2 is removing the non-nucleic acids. That's like scooping out all those annoying packing peanuts, the proteins and lipids. Step 3 is purification and binding, where we actually grab our prize. And finally, step 4 is concentration, which gets our DNA ready to be used in the real world.
Moving on to section 2, the chemistry set, traditional lab methods. Okay, so how do we actually pull this off? Traditionally, this whole unboxing process relies on massive, centrifuge-heavy laboratories and, frankly, some very intense chemistry. To break open our microscopic box, scientists generally have to choose between 2 paths: chemical or mechanical lysis. Chemical lysis uses things like enzymes, detergents, and chiotropic salts. These essentially disrupt and unfold molecular structures to gently solubilize and, well, melt away the cell membranes. On the flip side, mechanical lysis uses sheer physical force. We're talking grinding, blasting the cells with high-frequency ultrasonication, or just literally crushing them with tiny glass beads. So it's really a choice between a gentle chemical unraveling or a pretty harsh physical shredding.
Okay, let's dive into this. 2 of the absolute most classic chemical extraction methods are guanidium thiocyanate phenyl chloroform and cesium chloride. Now, these methods are legendary for a very good reason. They yield incredibly high amounts of beautifully pure DNA or RNA. But there's always a catch, right? These chemicals are highly hazardous, extremely toxic, and in the case of ethidium bromide, they are highly mutagenic. Because of that, they require serious protective gear and some really heavy, expensive laboratory infrastructure just to handle safely.
Which brings us to section 3, the solid phase revolution, moving away from liquids.
Now, because of those dangerous toxic liquid chemicals and the need for giant centrifuges, the science just had to evolve. We needed a path to portability. Enter modern solid phase methods like silica matrices and magnetic beads. These completely change the game. Silica works beautifully by using positively charged ions to magnetically grab onto the negatively charged DNA. But magnetic beads, they take it to a whole other level. By attaching the DNA to microscopic magnetic spheres, we can just use a simple magnet on the outside of a test tube to literally pull the DNA to the side. This completely eliminates the need for bulky centrifuges, which is just a massive step toward equipment-free processing.
Next up is section 4, moving to point of care, miniaturizing diagnostics. You see, getting rid of all that heavy equipment really is the ultimate goal here. We want to take these incredibly complex, bulky lab processes and make them accessible anywhere in the world. This is where point-of-care diagnostics, or POCDX, comes into play. It is a total paradigm shift. We're talking about moving away from massive, multi-million-dollar clinical laboratories and replacing them with cheap, portable, microanalytical devices. Just imagine taking an entire room full of spinning centrifuges and toxic chemical hoods and shrinking the whole thing down into a tiny plastic cartridge you can comfortably hold in the palm of your hand.
As this quote perfectly captures, the core ambition here is to replace those massive machines and operate near the patient at the point of care. And why does this matter so much? Well, because in a medical emergency, or say in a remote village without access to major infrastructure, you simply don't have days to wait for a lab result to come back. Empowering patients and doctors with rapid, on-the-spot healthcare decisions absolutely saves lives.
That leads us into Section 5, when chemistry melts plastic and engineering roadblock.
But as you might guess, this stream of palm-sized diagnostics hit a massive, totally unexpected roadblock. So the crucial point is that making things smaller simply isn't enough when your core chemistry literally melts its own container. It turns out the harsh chemicals that are absolutely essential for breaking open cells and purifying DNA, stuff like phenol and chloroform, are fundamentally incompatible with the cheap disposable plastics we use to actually manufacture these portable cartridges.
Now, what's really interesting about this slide is just how total this incompatibility is. When standard laboratory chemicals like phenol and chloroform meet common manufacturing plastics like PDMS, PMMA, or polycarbonate, which are all standard types of clear polymers, it is a complete and utter failure. Across the board, they are simply not compatible. The very chemicals we rely on in the lab actually act as destructive solvents the second they're introduced to a portable plastic microchip.
And let me tell you, the destruction is severe. When these chemicals touch the plastic, we see major structural consequences like depolymerization, swelling, and severe stress cracking. But perhaps the most devastating issue for diagnostics is the loss of transparency. You see, a lot of these point-of-care devices rely on lasers or ultraviolet light to literally read the final DNA result. If the clear plastic, like PMMA, becomes cloudy or completely opaque because the solvent melted its surface, well, the device is completely blinded. The test fails. Full stop.
Finally, let's look at Section 6, the future of diagnostics, overcoming limitations.
To resolve this incredible engineering puzzle, chemists basically had to go back to the drawing board and develop brand new chemical substitutes, ones that can gently extract DNA without eating right through a plastic cartridge. And to guide this whole future, the World Health Organization created a brilliant checklist known as the Assured Criteria. For any point-of-care technology to truly succeed on a global scale, it must be affordable, sensitive, specific, user-friendly, equipment-free, and deliverable. And the amazing news is, we're getting so close. Things like microfluidic paper-based devices and those new magnetic bead technologies we talked about are paving the way to hit every single one of these marks, all without melting the tech.
I want to leave you with this thought today. If engineers can fully solve this microscopic mystery, if they can perfectly balance that harsh extraction chemistry with the right portable device materials, what will universal, equipment-free diagnostics actually mean for our world? It means unlocking personalized medicine, instant outbreak detection, and life-saving diagnostics that are accessible to every single human being on Earth, regardless of where they live. Now that is a future worth engineering. Thank you so much for joining me on this explainer, and keep exploring.